Hi everyone, welcome back to my dodgy at home lecture filming process. So whilst the audio on Schoology is still being a bit hit and miss, this is kind of the best way I can think to go through this kind of bits and pieces with you guys. So a couple of things. First one is the mind maps or notes that I'm using to kind of talk through with you guys that will be available for you on Schoology. So if it's not quite visible on the video, or it's not zoomed in enough, or whatever it might be, then don't stress too much about it. You can follow along with me and then see those notes separately. Second thing is, I apologise if there are dogs barking in the background. I'm at home, the dogs are at home, so it might happen. So to begin with, we're kind of starting our chunk about photosynthesis, my favourite. So the first things we kind of need to talk about are the chloroplast and the chlorophyll. So we're going to kick off with the chloroplast first of all. In the top left hand corner up here, you'll see my dodgy drawing. I've kind of attempted to draw the diagram that's on page 8 of the textbooks. I know you don't have your textbook, but I have put a PDF up of these pages so you guys can see what I'm referring to. The diagram is kind of useful to have in your head because I think it just kind of helps you make sense of the different structures, okay? As well as recognising this kind of cartoony type diagram, you guys also need to recognise what a chloroplast looks like under EM, so electron microscopy. So we will have done a little bit about that last year, I believe, when we were looking at kind of electron microscopy. But as a refresher and a reminder, again, page 8 of your textbook, there is an example. Okay, so you need to be able to kind of pick it out of a lineup type thing. Now, on average, a green plant cell is going to contain 10 to 50 chloroplasts. There are some exceptions to that rule. Your root cells, for example, aren't going to have a lot of chloroplasts for what I hope is quite an obvious reason, given that they're not exposed to sunlight and they're not going to be actively photosynthesizing. And then that chloroplast, there are various different structures that you kind of need to be able to pick out and label and describe the function of. So in no particular order really. The first one is the chloroplast envelope, which is this inner and outer membrane and the space in between them, essentially, okay? So that's what your chloroplast envelope is. You then have the grana or the granum, different between singular and plural, and these are the system of membranes inside the chloroplast arranged in layers. If you have a look at that picture, as I said to you on page 8, it's kind of slightly more obvious than in my dodgy drawing. Now, those grana are made up of these thylakoids, and these are these individual discs, and these are membrane discs, and they kind of stack one on top of each other to make the granum, or the individual grana, and this is where that chlorophyll, that green pigment, is found. We then have the lamellae, excuse me, and these are the extensions of these thylakoid membranes, so these kind of bits here that I've got drawn. And they connect two or more of the grana, and they basically just act like a skeleton, and they help to maintain the distance between the grana, so they kind of help with the orientation of them, so they don't kind of move around and bump into each other. And the reason why you want to keep them in a particular place is it helps you maximise the amount of light they're being exposed to, and therefore the amount of light that they can absorb, and the amount of photosynthesis they can carry out. So we kind of need that structure, that lamella, that kind of skeleton in there, just to help keep everything in place. And then lastly, but by no means least, we have the stroma, okay? And that's essentially the matrix that surrounds the membrane layers, so kind of like the background kind of stuff, and it contains the photosynthetic enzymes. When we look at photosynthesis in more detail over the next few lessons, you will get to know those enzymes, but you should be aware that photosynthesis is a kind of enzyme control reaction, and so the stroma is where you'd find those enzymes hanging about, ready to do their job when photosynthesis occurs. Now, from GCSE, you guys will know and will understand that chlorophyll is the light-capturing photosynthetic pigment. So it's there to help capture and absorb that light. And remember, plants turn that light energy and use that light energy to power the photosynthetic reaction. When we talk about it at GCSE, we kind of just talk about chlorophyll, and perhaps it's kind of accepted by you guys that we're just talking about one molecule, like you might talk about water, for example, but it's not, okay? Chlorophyll is actually a mixture of closely related pigments. So within a plant, you will find I'm sorry, my two dogs are playing. Yeah, within a plant cell, within that chlorophyll, you would find several different types, okay? And they kind of mix together in different amounts to produce what we would call chlorophyll in general. So the different pigments are chlorophyll A, chlorophyll B, the carotenoids, and I've got to remember how to say this one, phaeophytin. Okay, now chlorophyll A is blue-greeny in colour itself. Chlorophyll B is yellowy-green in colour. The carotenoids are orangey yellow, and then the phaeophytin is actually grey, but this is a breakdown product of the others. So when the other ones are broken down in different chemical reactions, this kind of grey phaeophytin is produced. Now chlorophyll A, which is the blue-green, is found in all photosynthesizing plants, and it's in the highest quantity. Okay, that's why the vast majority of our leaves look green, because they've got this, their biggest amount of pigment is in chlorophyll A. Different plants will have slightly different amounts, but chlorophyll A is always going to be the most predominant one, and then the other ones are going to be present in various quantities. And that's what gives you the differentiation in colours of leaves in different plants. So when we look at a leaf, typically all leaves look kind of green, but if you examine them closely, you'll see they're slightly different. Aquatic plants, for example, so ones that live kind of under the water, might look slightly more bluey, or not slightly more bluey, sorry, they'll look slightly more browny reddy in their colour, because of how they've got different arrangements and amounts, volumes of the different pigments. Okay, so each of the pigments absorbs and captures light from particular areas of the light spectrum. So as you guys will know, white light as we see it is made up of 7 different types of light, and they have different wavelengths. Okay, and so each of those pigments is going to absorb light from a different wavelength. So the advantage to a plant of having a mix of these different pigments as opposed to just one singular one is that much more of the energy from the light falling on the plant can be used. If you imagine, light from the sun is white light, okay, so that means that all those different wavelengths are falling on the leaf. If you've only got one pigment, then you can only absorb one particular wavelength. So potentially you're going to be wasting, 80%, let's say, of the potential energy that's falling on your leaf. So as a plant, that's not particularly efficient when you're reliant upon that light capturing and that light absorption in order to kind of generate your energy and generate your glucose for food. Now, there are a few different ways that we can demonstrate these different pigments, a few different ways that we can show them, and we can do some work around them. The first of which is an absorption spectra. And this describes the different amounts of light of different wavelengths a photosynthetic pigment absorbs. So if you were to look at it on a graph, on our x-axis you've got the light wavelength, and that would be measured in nanometers, and you'd have a range of numbers. And then on the y-axis you've got relative absorption of light. Okay, again this graph is on page 8 of the textbook, and it's drawn slightly better than my scribbled version, but as you can see we've got different coloured lines which represent the different pigments. Okay, so towards the lower end of the wavelength measurements, a particular pigment is more effective, and then it changes as you move up. And you can produce absorption spectra either for the individual pigments, which is what this graph here would show, or for the chloroplast as a whole, assuming all the pigments are combined. So you could have the light wavelength and the absorption of light, and you just have one line that would vary up and down, kind of taking into account all of the pigments in one. Okay, so absorption spectra, we are measuring the absorption of light by the chlorophyll at different wavelengths of light. Okay, we then have an action spectra, which is slightly different, and this compares the rate of photosynthesis with the wavelength of light. So we're not talking about the absorption of light, we're talking about the rate of photosynthesis. Now Engelmann is a scientist, and in the 1800s he kind of first did the first action spectra. And how he did it is he placed strands of algae in the light of different wavelengths, and then he used bacteria that moved towards oxygen to show where the most was released. So the idea being that if, for example, the bacteria moved most to the wavelength of around 500, then he knew that must be where the most photosynthesis was occurring, and therefore that was where there was kind of the most action going on, so that would have the highest rate of photosynthesis. So a graph for that might look something like wavelength on your x and then number of bacteria on your y. Now, modern action spectra don't use those bacteria. Instead, they can use electronic data logging. So the idea would still be the same. You're still going to be comparing your rate of photosynthesis, and the most common way of measuring rate of photosynthesis is to look at oxygen production. Okay, and then lastly we've got chromatography. I haven't mentioned a lot about this because it's kind of difficult to explain in any other way that's different from what's in the textbook. Plus, I'm going to assume that most of you will have come across this at GCSE Science. If you're not sure what chromatography is, have a quick Google, well not a quick Google, but have a quick look on YouTube. There are loads of videos that discuss it to kind of show you the technique behind it. But essentially you can use chromatography and you can measure the RF values, and in measuring the RF values you will start to realise that there are those several different pigments that make up the entire of chlorophyll, and it's not just one pigment. Right, last little thing just to kind of tag on at the end, and I didn't want to put it on a new sheet because it seemed like a waste of paper, but just to make you guys aware, the photosynthetic pigments absorb light in two distinct chlorophyll complexes. So you've got these two different complexes, these two different combinations of your photosynthetic pigments, and they absorb light in kind of different ways or at different wavelengths. So photosystem 1, you have a combination of chlorophyll pigments which absorb light at the 700 nanometer wavelength, and that system is involved in cyclic and non-cyclic photosynthesis. When we look at photosynthesis later, you'll understand what that means. Okay, for now, just accept there's two different phases. So photosystem 1 or PS1 is involved in both, whereas PS2, photosystem 2, is a combination of those chlorophyll pigments, and they absorb at a wavelength of 680 nanometers, and they're involved in the non-cyclic only. Okay, so PS1 particles, sorry, I'm just trying to remember which way around it is, but PS1, when we've looked at electron micrographs, we have seen that the PS1, the photosystem ones, are mainly located on the lamellae, whereas PS2 are on the grana themselves. Okay, and so it's just to make you aware that there's different kind of combinations, different complexes of these chlorophyll in slightly different locations in the chloroplasts. They absorb slightly different wavelengths of light, and they're involved in slightly different stages of photosynthesis. Okay, so that's that. Once again, I apologise for my very, very noisy dog. I'll try to do more recording at school instead of at home, but please let me know if you guys have any questions.